Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. This is the 9th episode of Season 5. Today I'm delighted to welcome Dr. Libby Cieniewicz, associate professor of plant virology in the Department of Plant and Environmental Sciences at Clemson University, Clemson, South Carolina. Dr. Cieniewicz also manages the Clemson Clean Plant Center associated with the USDA National Clean Plant Network. Dr. Cieniewicz received her BS in biology from Lebanon Valley College in Annville, Pennsylvania in 2014, and her PhD in plant pathology and plant microbiology from Cornell University in 2019. She joined Clemson's faculty in 2019 as an assistant professor and was promoted to associate professor with tenure in 2025. Dr. Cieniewicz teaches courses in scientific writing, plant virology, and molecular plant microbe interactions, among others. She has received outstanding teaching awards in 2024 and 2025 for her efforts. Dr. Cieniewicz has a diverse and robust research program which aims to understand plant virus ecology and epidemiology, interpathogen interactions, and co-infection dynamics, as well as develop innovative methods for virus diagnostics and management. She's co-authored more than 24 papers, largely focused on insect-vectored viral diseases impacting fruit. Beyond research and teaching, Dr. Cieniewicz contributes widely to various university committees and professional societies, including serving as a faculty senator at Clemson and an advisory member of Plant and Pest Diagnostic Clinic there. She served as a subcommittee chair for the committee to develop a full list of plant pathogens for USDA APHIS, BRS, and APS, and has served as a senior editor of APS's Plant Disease. Welcome, Libby. We haven't had many plant virologists joining us here on the podcast, so thanks for joining us.
Yeah, thank you for the invite, Matt. I'm happy to be here.
You recently received tenure, is that right?
Did in May. Yes, I got the letter on May 1st, and then I went into labor like a day later, and I had my daughter on May 4th.
Congrats to both of those things. That must have been a hell of a week.
It was interesting, yes. Thank you.
As a plant pathologist who studies fungi and teaches general plant pathology, I find viruses to be quite enigmatic, even for students. Now, this is interesting because if you ask anyone on the street if they know what a virus is, or if they could give an example of a virus, at least a human virus, everyone would be like, oh yeah, I know viruses, and they list off flu or COVID or measles or RSV. Yet, if you ask someone to define what a virus is and how it looks or how it operates, not many people can answer that. I don't say that to belittle people. Viruses are strange, and I think it would be helpful for the listeners if we could start off by defining what a virus is in terms of plant viruses so that the listeners that may not have a background in plant pathology understand.
Yeah, so that's actually how I start off. I teach plant virology in spring semesters. I teach it every other year, and so I'm teaching that this coming spring. And I have undergrads and graduate students in this course, and that's actually how I start off the class every year, is with the actual textbook definition of a virus. And so I use the Roger Hull plant virology textbook. It's like the main plant virology textbook available. So I use the definition from that textbook, and we break it down. So there are like a few parts of the definition, like it's small. We have the debate in my class of whether they are living or non-living. So that's a whole nother interesting thing we could get into, but I usually don't give that one away right away. I let the students decide that for themselves. And then really the definition of a virus, what makes a virus is that it has a genome and a capsid structure. But recently, I think we're seeing those rules or definition bending a little bit too. Yeah.
So I typically define it for my students as an infectious nucleic acid or a parasitic nucleic acid. Obviously, it's able to self-replicate inside a host, but it doesn't respire, it doesn't move, it doesn't respond to stimuli. But it's really interesting because that whole debate of whether or not it's living or not, I think a lot of people, when they think about viruses, they think about bacteria or fungi. They're like, it's a small microorganism that moves around and does what it wants. But that's not really the case with viruses.
Yeah, and defining them by their size, I don't think is really effective anymore because there's these giant viruses that have been discovered in the ocean, and those are making a gray area between what is a virus and what might be a bacteria or some other type of organism.
Yeah, and on that topic, I know that historically the shape of the virion in the arrangement of the capsid, as well as the type of nucleic acid inside, obviously dictates how we categorize them. Is that right?
Yeah, that's right. So viruses are typically categorized by either the type of genome and or the capsid structure. But really going back to that definition of is it living or non-living, that kind of question that we discuss in the class. It's a fun discussion, but it also changes the words that you can use to describe viruses, right? We say that viruses replicate, not that viruses reproduce. Or we say that they persist outside of a living host rather than they survive outside of a living host, right? And so it changes the words that you can use.
Because it's not like they're inert outside their hosts, correct?
Yeah. So I'm giving away the answer to my question. I think they are non-living biological entities, but
Yeah. I think that's important for our listeners here. Now, viruses also have many disease vectors, including insects, mites, and nematodes that aid in transmission. And there's different types of transmission. There's stylet-borne and circulative and propagative, and we could talk a little bit more about that. But a more basic question is, are most plant virologists, entomologists, and or nematologists by necessity?
I don't know if most of them are. That has been my experience. So I had no interest initially in entomology, and I don't actually even have any formal training in entomology, but now I'm able to advise PhD students in the entomology program at Clemson. So who am I to be an entomology program advisor? But really, I gained my entomology experience through my research and again, by necessity of the virus that I was working on in my graduate work.
I would actually say minoring in entomology is a qualification, but
I guess it is. Yeah, but there wasn't really any formal coursework that enabled me to minor in entomology.
sure. Coming back to the insect vector part, though, I think they're useful for several reasons in plant virology, and I'd like to know your opinion. I came up with 3 reasons, and it's not comprehensive, but it's just off the top of my head. It gives the virus a face in the absence of an electron microscope. It gives you a target for control, and it gives you a target for early detection and rapid response. Would you agree with those 3 criteria?
Absolutely. All of the above. I think you can make those statements. Maybe your number 2 about an option for management. It's definitely more nuanced than that. And that's where some of my work has been aimed at is trying to determine if and when targeting the vector will be actually effective in stopping the spread of a virus disease. So my best example that I can think of is the virus that I worked on in my PhD, grapevine red blotch virus, is vectored by a tree hopper. And that's such an interesting system ecologically because the tree hopper vector is not really a pest of the host crop, grapevine. So it doesn't really like to feed on grapevine. It's weird, right? Vector's the virus, but it's not really preferring to feed on grapevine. And so the work that we've done over the years has led us to the conclusion that targeting the vector in that case isn't really likely going to be effective because populations are already small. So if you reduce populations of the insect vector in that case, it's not going to hurt, but it's probably not the management tactic that's going to result in the slowest spread. So really the data shows that you should be targeting the initial inoculum levels and like spread of the virus through infected planting material. Another example is in the work that we're doing here at Clemson with some peach viruses that are transmitted through pollen. And so their vectors are bees. And
Oh,
so
wow.
we're not gonna kill the bees, of
Sure,
course.
that
So
makes sense.
yeah.
Now, one of the control strategies I've heard for in plant viruses, and it may be specific to certain types of plant viruses, but this idea of cross protection where you use a mild strain to protect against these more virulent strains. Is that something that's commonly still used across, like broadly in plant virology?
I think it's something that we always like want to consider, but it relies, of course, on having identified strains that are inducing mild symptoms. So there are some products. I actually teach about this in my class. There's a product for Pepina mosaic virus that's used in like greenhouse production. I think it's more commonly used in Europe than in the US. but it's for Pepino mosaic virus in greenhouse settings. Another place that cross protection has been applied is in citrus for citrus tristeza virus.
Okay, that makes sense. And otherwise, this virus-free seed and surveillance are the best strategies.
Yeah, again, it depends on the system, I think. That's where a lot of the focus is for my lab's current research. And we operate the Clemson Clean Plant Center. So we work with the National Clean Plant Network. The purpose of the Clean Plant Center is to provide sources of peach tree material that are negative for all the known viruses. Again, because most of the viruses that we're concerned about in peach are transmitted through pollen. So there's nothing you can really do to reduce spread once the infected trees are out there. So really the only point where we have control in the system is before planting and during propagation.
Does that include Prunus necrotic ring spot virus?
Yeah, that's one of them. So the two that are transmitted through pollen are Prunus necrotic ring spot virus and prune dwarf virus. And those are both in the Eiler virus group.
Do nematodes also play a role in transmission of that virus?
Not for those two viruses. No.
Okay.
So there are some nematode transmitted viruses that are also transmitted through pollen. So some of the Nepo viruses like tomato ring spot, tobacco ring spot virus, those are transmitted both through pollen and through nematodes. So that complicates management for a lot of viruses where there's multiple transmission modes. I've always said it's important to know like what the possible transmission modes are and really doing like the replicated lab studies and greenhouse studies for knowing how viruses can be transmitted is one part of the equation. The other part is, okay, what's actually happening out in the field? So that's where a lot of my research lies.
That's great. And thanks for the clarification there. I want to come back to this grapevine red blotch virus that you specialized on for your PhD and continue to work on. We were talking about vectors and the role of controlling vectors. And I think where it becomes complicated, and you already agreed to this, is that you take a virus that has a pretty limited host range, like the grapevine red blotch virus, and then you look at its vector, the three-cornered alfalfa hopper, and that feeds on alfalfa, soybean, cowpea, sweet clover, in addition to grape. And that's really where the math doesn't add up for trying to manage a vector that has so many hosts that are separate from grape, especially if it's not visiting grape very much.
Yeah, the math is not mathing there.
Exactly.
And that's been one of the big questions with red blotch once the vector was reported. So the virus was reported in 2011, 2012, and then the vector was reported in 2016 from a group from UC Davis. So it was a little bit of a race to figure out what the vector was. And once the vector was identified, then it was about, okay, that doesn't really make sense. That's not even a pest of grapevine. What's it actually doing in vineyards? So then a lot of the work that followed the vector discovery was, okay, what is this tree hopper doing in vineyards?
Okay. To take that one step further, how common is it for viruses to have multiple species or genera of insects serving as vectors? You mentioned CTV, the citrus tristeza virus. Now I know that has the melanin cotton aphid, the green citrus aphid. It's got the brown citrus aphid, the oriental citrus aphid. Those are different genera, multiple species of two different genera. That really complicates things, especially when you're getting introductions of novel vectors. How common is that in plant virology?
I think the introduction of novel vectors is very common, and that explains a lot of the range expansions of virus diseases as well. I think it's less common for a virus to be vectored by multiple insects. Of course, there are examples. So another grapevine example is the leaf roll viruses. So grapevine leaf roll virus 3 is the most important leaf roll virus of that complex, and that's vectored by several species of mealybugs. Of course, at different efficiencies, and the mealybugs have different geographic ranges as well. So, yeah, that's where the ecology work really comes into play, right? It's important, of course, to do the replicated studies and know what viruses can be vectored by what insects, but then you also have to, I think, take it one step further out to the field and really figure out what is actually happening. So what can potentially happen is a question that you can get at more in the lab, but what's actually happening out in the field, you have to go put your boots on and go out and get dirty.
Yeah, absolutely. I think on your web page, you quoted J.C. Walker about having 1 foot in the furrow.
Yeah.
So I agree with that. One of the other things that complicates this is that some of these hemipteran insects, we have introductions of novel species, but even the taxonomy of native species is under revision. So we may think we have one species of insect vector, but it's really a species complex, and their contributions to contributing to disease dynamics may differ largely. So that's another complication, right?
Yes, yes, it adds like another node to these complex issues.
Now, you studied biology as an undergrad at Lebanon Valley College in Annville, Pennsylvania, northeast of Harrisburg. Are you from southeastern Pennsylvania, and what got you interested in biology and ultimately plant pathology?
Yeah, yeah, so I'm from Berks County, so it's like the next county over from where Lebanon Valley College is, and I grew up with a general love of the outdoors, but like most high school students who are interested in biology, the only thing I was really aware of that I could do with biology was pre-med. So that's how I started my degree at LVC. And then, so the professor who really changed my track at LVC, her name's Dr. Becky Urban, and she recently started an environmental sciences program at LVC. So at the time I was there, it was just biology. And I was able to take some courses in plant biology, but in my coursework at LVC, I really started to get interested. I loved microbiology and I really loved the plant bio. And at the time, I didn't even really connect those as, okay, plant pathology is what I want to do. But it was the summer after my sophomore year, I applied to 2 research internships, one at the Penn State Hershey Medical Center. I was interested in like neurobiology at that point. And then the other one I applied to was the Cornell Summer Scholars Program in Geneva. And I ended up not getting into the one at Penn State, and I got into the one in Geneva. And I mean, the Summer Scholars Program is totally the track that I went down then, and it changed my life.
Yeah.
It's like the old one door closes, another door opens
Yeah.
thing. But
Yeah. We're all where we are because someone said yes at some point.
anyway, yeah.
So viruses are pretty small, although, as you said earlier, that there are some large viruses in the ocean, and we're just learning about them. Tobacco mosaic virus is really important in all virology, not just plant virology, because it was the first virus discovered and crystallized, and it's a single-strand RNA virus. It's about 6,400 base pairs long, 158 amino acids. Are most plant viruses roughly similar in size? Because I think about fungal genomics and how we can have a 1.5 gigabase genome, but then we could have a 10 million base pair genome. Oh, is there a lot of variation there in plant viruses?
Yeah. How long did you say the genome is for
Well,
TB?
TMV is 6,400 base pairs.
That's about average, I would say, maybe on the small side. Now, the virus genome structures are very simple. So they basically encode for 4 genes. And you do get several viruses that even if the genomes are smaller, they might be a little bit more complex in terms of the genes that they encode. So a good example of that is the Gemini viruses, like grapevine red blotch virus that we've been talking about. That one has a very small genome, like 3.2 kb, so about half the size of TMV, but it encodes at least 7 orfs, so a little bit more complex. And that's part of why I love viruses, is because I'm going to personify them, but they're very clever in how they express their genomes, and they don't like to carry a lot of baggage, but they are very efficient in how they get around and do what they want to do.
Yeah, they sure know how to pack a punch, don't they?
Yeah.
Now, you've devoted a considerable amount of your career to the grapevine red blotch virus that we've been discussing. Did your PhD at Cornell in Geneva, in one of the most important grape growing regions in the East, the Finger Lakes. It's not hard to sell what you do to the general public in those areas. Now you're in South Carolina, where soybeans, cotton, peaches, and peanuts really represent the most important economic crops. Do you see yourself drawn to more regionally important plant viruses? And before I let you answer, I'm going to say, I know that going into this, that muscadine varieties of grape are susceptible, or at least there's grape varieties that grow in South Carolina. And I also note that you've worked on Prunus necrotic rain spot virus, peach latent mosaic viroid, cucurbit leaf crumple virus, and cotton leaf roll dwarf virus. So clearly you're invested in the crops that are important to South Carolina, but talk more about that.
Yeah, so when I started here at Clemson in 2019, I started really right away with working with the peach industry. So there was a long-standing program it's called the Southeastern Budbrood Program, started by my predecessor, Dr. Simon Scott, in 2001, I think. And again, it has to do with the Clean Plant Center work that we do, make sure that the material that's being propagated is negative for those viruses. But we've done some biology and ecology research on Prunus necrotic ring spot and Prunendorf virus, and most recently this viroid in peach-light mosaic viroid. So we started testing for that in 2023 and found out that is basically all over the place. Growers are really concerned about it. So a lot of my lab's work right now is focused on peach-light mosaic viroid, and kind of what we were talking about earlier, like several modes of transmission have been reported in the literature, like in greenhouse studies. For example, we know that it can be transmitted through pollen by the green peach aphid, Mysis persicae, and mechanically, like on contaminated tools. So we know from greenhouse studies that all of those things can happen. What we don't know is, are those things actually happening in the peach orchard? So we have some research going on right now to understand how the viroid is spreading in the peach orchards.
Now, right before this, we were talking about virus size, and we're now talking about something called viroids, which are slightly different than viruses. Again, always to educate our listeners, how does a viroid differ from a virus?
Viroids are so cool. So they are not viruses. They are sometimes referred to as subviral agents, and they're very small. So the one peach-light mosaic viroid is like 340 base pairs, and they are just these tiny, naked, circular RNAs, and they form this really cool secondary structure in the genomes. But it is truly amazing to me how these tiny pieces of RNA that they don't even code for any protein. They don't even code proteins. They're just infectious RNA molecules, how they are stable in the environment, and how some of the symptoms that these viroids can cause are just incredibly dramatic. So one of the examples that I use in my class is the coconut kadang viroid. The
love that one because it's the best name ever.
Yeah, it's awesome. Viroid diseases have some of the best names.
Yeah, like
There's one that's a scaly butt of citrus.
yeah, and apple dapple and apple dimple, right?
Yeah, apple scar skin. Yeah, there's really cool names for viroid diseases.
Okay, so you say you obviously involved with the peach industry, which is an important agricultural crop there in South Carolina and throughout the South. Now you've worked with this cucurbit leaf crumple virus. Presumably, cucurbits are among the hosts there. You never know with these common names, but what does that do and what's its impact there?
So that was just one paper that I worked on, that I was a co-author on for that virus. It was a diagnostic assay. And that's another big area of my lab, is optimizing and developing new diagnostic assays for viruses and viroids. So there's a virologist I'm not the only plant virologist in South Carolina, unfortunately. But yeah, so my colleague, Shu Ling, is at the USDA Vegetable Research Lab in Charleston. And so he covers most of the cucurbit and tomato and like most of the vegetable viruses. And really, I've been focusing more on the fruit crop viruses. But there's a lot of emerging cucurbit viruses. There's a working group that is composed of people from all over the US who are focusing on these emerging cucurbit viruses. And what we were talking about before with, like, when a new vector comes, then it expands the range for that virus. Or we've seen this, of course, like with citrus greening and some other vector-borne pathogens, where the vector expands its range, the virus tends to follow, or vice versa. So yeah,
So,
that's a big thing with the cucurbit viruses right now.
okay. So tomato ring spot virus is a broad host range virus that impacts a number of things, including cowpeas, which are South Carolina's the number one producer. But it sounds maybe those are not impacting. You're not working on that virus, correct?
Not really actively. We've found it. We did a big survey of blackberries. So blackberries and berry crops are like gold mines for virology. There's tons of viruses and new ones being discovered all the time. Yeah, we did a recent study in blackberry where we sampled from 8 farms around the state. We discovered several novel viruses and found lots of known viruses as well.
Okay. Do you want to tell us a little bit more about your role there in the Clean Plant Center? You told us that this is like you're part of this Clean Plant Network, which is a USDA network. And tell us a little bit more about your role there and, how much diagnostics you're doing, what your turnover is, like how many clean plants are you producing a year, all that.
Yeah, sure. The NCPN was formed along with the Farm Bill in 2008. But The NCPN didn't create the Clean Plant Center, so lots of the Clean Plant Centers and Clean Plant programs around the U.S. And I should say first that the crops that are covered by the NCPN are the vegetatively propagated crops that have virus issues. So it started off with grapes and fruit trees, but now it's expanded to grapevine, fruit trees, sweet potato, berries, citrus, hops, and roses. So it's an interesting group of crops. But the NCPN is housed under APHIS and provides funding to our program every year. But my program, which in 2020, we renamed it the Clemson Clean Plant Center. And so it's just part of my lab where, again, our main goal is to provide sources of virus negative material for the peach industry. So we're part of the NCPN fruit tree group. The other centers that are in the NCPN fruit tree group, there's one at UC Davis. So it's Foundation Plant Services. They're the biggest clean plant center in the US. And then the other one is the Clean Plant Center Northwest at Washington State in Prosser. So they handle the West Coast, obviously. We're holding down the fort here on the East Coast. And the goal is to make sure that essentially all the new trees that are being planted in the Southeast are negative for the viruses that are going to be causing problems. And so the testing program was started by my predecessor, Dr. Scott, in 2001. And this was for a couple of reasons. In the late 90s, they had done some surveys and were seeing some issues with PNRSV and PDV, so the two Eiler viruses that are transmitted through pollen. And through these surveys found that the incidence was pretty high, so they wanted to address that problem. But also, plumpox virus was discovered in Pennsylvania and then in some of the other Great Lakes states in the years following. And so that kind of scared everybody, especially in South Carolina, where peach production is really concentrated in a couple of areas in the state. And because if plumpox virus were to be discovered in South Carolina or in Georgia, it would be a big problem because there's harsh eradication procedures that would have to be put in place, and it would really negatively impact the peach industries. So we still test for plumpox, even though it's never been found in the Southeast, because never say never. And my first year doing the testing program was 2020. And obviously 2020 was a crazy year for viruses. And so I was like, man, if we're going to find plumpox in South Carolina, it would be in 2020. We did it. So good news. But we continue to be vigilant for that reason. And so the way that the Clean Plant Center operates is that we have three main activities that we do. We do virus diagnostics. So this is on plant material that is going to be used for propagation. So if we have a breeding program for peaches at Clemson, and anytime that there are new varieties released from either the Clemson breeding program or USDA breeding programs, we want to make sure that there are nuclear stock materials that are negative for the viruses and are clean, so that we can always go back to those to propagate new plants for the industry. So anytime there's these new releases, we do diagnostics. So we use high throughput sequencing to detect any and all viruses in those samples. And then if they are infected, then that goes into our second activity, which is virus elimination therapies. So this is a lot of protocol development. We're actually really actively right now trying to figure out how we can remove peach latent mosaic viroid from peaches. It's really hard to remove because it invades the meristem. And so viroids are really difficult to eliminate from plant material. But then once the plant accession is clean, then we maintain it in what we call a foundation collection. And it's similar to a germplasm collection where we keep 2 or 3 copies of each tree under conditions where it's protected from reinfection. It's an insurance policy, but also functional in the way that we can provide that material to the industry to propagate the new trees.
Thanks for that. That's really interesting work and really important work. I'm going to come back to the plumpox virus because I'm from Pennsylvania. I did my PhD at Penn State, and Fred Gildow, who was one of my professors at the time and eventually department head, was heavily involved, I think, with the eradication programs there. I want to come back to the point that, 2020, there was testing for plumpox. It was only recent, like in the recent, last 5 years, that it was declared successfully eradicated, correct?
Yeah, I think they had a big party in DC. It was 2019 when I started my position here that it was declared eradicated from the US.
And it was discovered in 1999, or at least the late 90s. And that just goes to show how long it takes to successfully eradicate something, and only under extreme quarantine conditions and destruction and a lot of funding through the federal government to subsidize a lot of the destruction of these entire orchards.
Yeah, a lot of funding. And the lab that I did my PhD in with Mark Fuchs at Cornell, so they did I think they still do it, probably not to the level that they once were, but at one point they were testing like 130,000 samples a year, stone fruit samples a year for plum pox. So they continued to do these surveys for years and years, and that was run through the state departments of ag. So Pennsylvania Department of Ag had its own surveys going, and then this was the New York Department of Ag and Markets that was funding this work through Mark's lab to do that testing. But I always joked that I would go downstairs to the lab where they were doing the testing, and they had it all 130,000 samples in a year. That's crazy.
Yeah.
So they had to be really efficient, and they would hire this army of like undergrads and high school students, and it looked like Charlie and the Chocolate Factory, like looking for the golden ticket for Veruca.
It worked though, so that's great.
Absolutely. It's a big success story for eradication.
I want to come back to this. It's kind of like the basic biology of viruses and talking about disease vectors. There's viruses that are stylet-borne, which means that they're non-persistent and they are only on the piercing, sucking mouth parts of these various hemipteran hosts, or I guess they could be on nematode stylets too, so it's whatever the vector is. There's this circulative, which means that it has to work its way through the host and then back into probably the salivary gland and then back out the mouth. But they don't multiply inside that host. And then there's propagative or persistent. It seemed to me that stylet-borne would be the most dangerous because they're ready to inoculate as soon as they touch it. But maybe that's wrong. Which ones pose the greatest threat in terms of types of viruses?
Yeah, I feel like I give a lot of politician answers here. Like, it depends on the situation, right?
Yeah.
And it truly does. So talking about plum pox, I think one of the reasons why the plum pox eradication program was so successful, or why the problem wasn't worse, is exactly because of the transmission mode. So that is an example of a virus. It's a potivirus. So it's transmitted in a stylet-borne manner by aphids. So it's like the combination of the transmission mode and the ecology and landscape composition in Pennsylvania. So you're from Pennsylvania. It's not like there's just like acres and acres of stone fruit production all kind of concentrated in one area. It's more there's a farm that's growing peaches, and then maybe it has apples, and its neighbor is maybe it's forested land, or maybe then you have another apple producer nearby. So I think the fact that it was a stylet-borne virus actually prevented it from getting out and spreading really widely in Pennsylvania, because the minute that aphid picks up the virus from that peach tree and then it goes and feeds on an apple tree, the apple tree is a non-host for the virus, so it's like cleaning its stylet off in those non-hosts. Whereas if it was a virus that was transmitted in a circulative manner, then that insect vector is typically going to remain viriliferous or able to infect for the rest of its life. And so it would feed on that apple, but then maybe it would go to another peach tree and be able to infect peach or cherry or whatever. So the fact that it was stylet-borne, that makes it really able to transmit quickly and easily, but it also, then if it's feeding on a non-host, then it's getting rid of the virus. It depends really, like, you need to look at the full context of the landscape and ecology of these systems, I think.
And even if it doesn't feed on a non-host, it will only persist for a couple hours.
Right, yeah. Or as soon as the insect feeds, it's going to spit it out, and then it's no longer being retained. Another example of this is with a papaya ring spot. So I use these kind of case studies in my classes, and the papaya ring spot virus story is one that I think every plant pathologist should know about. But the transgenic papaya, the rainbow papaya, actually allowed for non-transgenic papaya to be produced in Hawaii for that exact same reason, where it provided a buffer zone, this herd immunity concept, where the aphid, even if it picks up papaya ring spot virus, because it's a potivirus as well, just like plum pox, it is going to then feed on the transgenic resistant papaya, cleanse its stylet, and then you have cleaner aphids flying around.
Right, that makes sense. So on the other side of the coin, in this persistent propagative, you have this latency period where the virus is replicating inside the host, but the host isn't going to be able to infect new plants because it has to work, the virus itself has to work its way through the vector, and there's going to be a period of non-infectivity during that time.
Yeah, the latent period. Actually, with red blotch work that awesome student, Maddie Flasco, did in Mark's lab after I had left, and she picked up on some of the stuff I was doing with red blotch. She found that it takes at least 9 days for that to occur with red blotch and the tree hopper vector.
Oh, wow.
That's a long latency
Yeah.
period. Yeah, it's
Yeah.
not typically that long.
We were talking about the patchwork kind of landscapes of Pennsylvania, and Pennsylvania is not unique in this regard, but certainly when you look at the multiple ownerships and management strategies of orchard owners and landowners, there's two things happening there, which I think lean on two of your loves. One of them is this kind of natural reservoirs. You think about cherry, for example, black cherry being in the forest, and so any other prunus species that are in the orchard, there could be this natural environmental reservoir in native cherry. But there's also this interpathogen interactions. Clearly, there's other pathogens that are impacting fruit trees. And I teach this in my classes, like no disease happens in a vacuum. We have multiple contemporary threats to crop production. You can have fungal, bacterial, and viral diseases all impacting trees. And how do you see viruses interacting with some of these other pathogen groups, and what are the biggest kind of synergistic effects of that?
Yeah, I love it. Like virus synergistic effects in virus co-infections is one interesting area.
We'll talk about that.
Actually, let me talk about a virus and bacteria example because we have an ongoing project on this. So I have a PhD student. Her name is Daniela, and she is working on this project where we're trying to understand an observation that I and my colleague Haha Wang at Clemson, so she's the bacteriologist, I'm the virologist, and it was in 2020, we observed these orchards that I knew were infected with Prundorf virus. And I was I was new to Clemson. I was trying to understand, like, what do these viruses look like in the orchard? What are the disease symptoms? And we found through our testing program an orchard that was 100% infected with PDV. And so I'm like, okay, I'm going to go there. I'm going to go look for symptoms. I went and looked at these trees and they're beautiful. Like, they're perfect. I'm like, what the heck? And so I asked the grower, I was like, do you notice any symptoms any other time of the year? Or are these trees behaving differently in any way? And he said, no, I don't notice any typical virus symptoms, but that block seems to be more affected by bacterial spot. It's a Xanthomonas pathogen. And I'm like, ooh, light bulb went off. And so I tapped in Dr. Wang and I was like, let's go check out these orchards because we knew that there were a few other blocks that didn't of the same variety that did not have PDV. So we went to look at them. Just like early observations corroborate what the grower was saying, where the ones with the lower virus incidence had less bacterial spot. And none of this is quantitative at this time. We're just looking at the trees. And then the trees that were infected with the virus seemed to have more bacterial spot. So we've followed up on this over the last few years, and it's not super clean, right? It's not really consistent that we can see this like tight association between virus infection and more severe bacterial spot. Like in some years it's worse than others or it's tighter than others. But when we do replicated studies in the lab using detached peach leaves, we are able to observe this consistently, that the virus infected leaves are, when you inoculate them with the Xanthomonas pathogen, then you see bacterial spot developing more severely and more quickly in the virus infected leaves. And actually, of course, I went to the literature and I'm like, okay, what's known about interactions between viruses and bacteria? There's almost nothing. So there's one paper on rice that was published in 2017 and maybe a handful of other papers where they've looked at virus and bacterial interactions. So it's a new area, like ripe for the picking for us because no one has really looked at this. And I think it could help to explain to growers like who are not convinced by the potential impact of viruses and they're not convinced that it's important to manage viruses.
Yeah.
We can make that really clear that, hey, virus infection may not be causing these devastating symptoms on its own, but bacterial spot's like one of the number one diseases of peach in the Southeast. And I'm like, if we show that virus infection predisposes those trees to more severe bacterial spot, then I think that's a really important message for the growers to understand, like the importance of clean plant material. So Daniela's work is morphed into trying to understand how the virus is changing the physiology of the plant to make it more susceptible or like a more favorable environment for the bacteria to develop. So stay tuned for
Yeah. No, that's a great example. And I think it comes back to this: give the virus a face or give the grower a reason to better respect viruses and their potential. So that
Yes,
was really
absolutely.
interesting. So you are president of the Student Association of the Geneva Experiment Station. I know as a faculty member at Clemson, you've been involved with the GSA there. In fact, you're awarded for being an outstanding grad student advocate in 2022. What benefits do you think students receive from their participation, involvement, and a grad student association? And what do you see your role there as?
I mean, I think it's honestly not dramatic to say it's a make or break. If you have a community that is supportive as a graduate student, then that can make all the difference for you. Research is hard, right? You have experiments that fail all the time. And so research is inherently something that is going to continue to beat you down until you get
Yeah.
those times of discovery, and the good times come too. But in order to like deal with that constant, I don't want to refer to it as failure, but that like constantly being like beaten down by your research or facing these challenges that are inherent to what we're doing in scientific research, you really do need a supportive community. And I had that at Cornell. Cornell was a great place to be a graduate student. And I had this awesome cohort of other students that started at the same time as me. I'm still friends with almost all of them. So I had a lot of support at Cornell and a lot of opportunities there that I realized were lacking for a lot of the students in the department here at Clemson. And so that was one of the first things that I tried to do when I was here. I wanted it to benefit the students that were going to be in my lab too, right? I'm going to be hiring students and mentoring graduate students in my own lab. So I want to make sure that they have the support that they need in the graduate program. But really for all the students at Clemson, I wanted just to see more support, and I wanted them to feel like they were able to like have their voices heard. Because if you have one student all by themselves like trying to make a change in the department or advocate for reduced fees or whatever they're trying to get across, that message is a lot stronger when you have 100 students saying the same thing rather than just one. I'll never forget it. We had our first meeting of the Graduate Student Association here at Clemson. It was right as the pandemic was taking off. It was in early spring of 2020. And there were students who it felt like a therapy session a little bit. Like the students came and they were in tears about like how they felt isolated at the beginning. And I was like, man, so this is really needed. Like we really need to be forming the students together and helping them advocate for themselves.
Yeah, and I know participation in our Plant Pathology Grad Student Association was really useful in seeing people let down their guard because grad school can be competitive too. And you need to see people not focused on work sometimes to be like, oh, we're going to come together and have a community potluck, or we're going to have a cookie contest or carve pumpkins.
Mm.
It's like you need to do those things because you know what? You're not just a researcher, you're a human who is like multidimensional, and you need to
Right.
lean on some of those other dimensions sometimes.
Yes, that reminder that you're like a whole person and not just your failed experiment is
Yeah.
important.
Now, you've graduated 5 grad students, 2 PhDs, 3 master's students, and currently have 2 PhD students and 1 master's still in the lab, at least what I can figure out. How are you approaching career mentoring given the shift in employment opportunities and such? I say this, I mean, I've graduated 4 students myself in the last 14 months, including 2 PhD students, and it's been a little bit heartbreaking to figure out, how to guide them into a career opportunity lately.
Oof. Yeah, it's hard. I don't envy the students who are graduating right now and looking for positions because it is like truly dismal right now, the outlook of job opportunities. The 2 PhD students that graduated in 2024.
4.
Yeah.
Yes,
Yeah,
2024.
one in spring of 2024 and then one in August. And they were both looking toward like academic positions. And that's something I frequently discuss that with them throughout their degree here. What are your goals? What are you interested in? And my only entomology student, Mandeep, he was always interested in academia, but he also didn't want to be closed off to other opportunities. And so he did an industry internship. He worked with Corteva for a summer while he was still a student. And then he came back and finished up and graduated. And through that internship, he realized, hey, I think I really do want to go do an academic postdoc. And he had a couple of opportunities for postdocs. They've all landed in good positions. One of my master students is already working with me. So he's already like a technician and he graduated in May. So he's still working with me. But he was looking at maybe positions with the USDA. And then obviously was like, okay, I'm slowing that down and I'm going to stay here where I have a guaranteed paycheck and we're safe for a while. I don't think I really have any great answers. I don't know the magic answer. If you do, I'd love to hear it. But
I don't. I think that coming back to one of the classes you teach a class in scientific writing, and I'm guessing that's more formal scientific writing. But one thing that's become very clear over the last few years is our need to better communicate what we do, not only through peer-reviewed literature, that's something that we've always done, but like science communication that's more outward facing and communicating the importance of what we do. Sometimes when you work in plant pathology and you're protecting a very important economic crop, it's easy. You could say, this represents the largest economic crop in the state. We're working to save it from disease X or insect Y or things like that. But some of the more basic science we do, I think we could do a better job at communicating why that's important beyond just to our academic journals, but working through our universities to do kind of extension like bulletins and things like that. And I know you've been involved with that too.
yeah, I think like learning and teaching students to articulate the value of their work in different ways is important.
Absolutely. I've really enjoyed this kind of whirlwind conversation, mostly whirlwind because I went all over the place, but you followed pretty well. So thank you so much for your time.
Thanks, Matt. It's been fun.
We've just heard from Dr. Libby Cieniewicz, Associate Professor of Plant Virology in the Department of Plant and Environmental Sciences at Clemson University, Clemson, South Carolina. As with all our episodes, be sure to check out the show notes for links related to Libby's research program. I'm Matt Kasson, host of Plantopia. Thanks for listening.